Chapter 12, Section 3. The eukaryotic cell cycle is regulated by a molecular control system. The timing and rate of cell division in different parts of animal or plant cells are crucial for normal growth, development, and maintenance. The frequency of cell division varies with cell type. Some human cells divide frequently throughout the life of the organism, such as skin cells in humans. Other human cells have the ability to divide, but keep it in reserve, such as liver cells. Mature nerve and muscle cells do not appear to divide at all after maturity. Investigation of the molecular mechanisms regulating these differences provides important insights into the operation of normal cells and can also explain how cancer cells can escape those controls. The cell cycle appears to be driven by specific chemical signals present in the cytoplasm. Some of the initial evidence for this hypothesis came from experiments in which cultured mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei. Fusion of an S-phase cell and a G1 phase cell induces the G1 nucleus to start S phase. This process suggests that signaling molecules present in the S-phase nucleus stimulate the fused cell. Fusion of the cell in mitosis, or M phase, with one in the interphase, G phase, induces the second cell to enter mitosis. The sequential events of the cell cycle are directed by a distinct cell cycle control system. Cyclically operating molecules trigger and coordinate key events in the cell cycle. The control cycle has a built-in clock, but it's also regulated by external adjustments and internal controls. A checkpoint in the cell cycle is a control point where stop and go ahead molecules will regulate the cycle. The signals are transmitted within the cell by signal transduction pathways. Animal cells generally have built-in stop signals that halt the cell cycle at checkpoints until they're overridden by go-ahead signals. Many signals registered at checkpoints come from cellular surveillance mechanisms. Those mechanisms indicate whether those key cellular processes have been completed correctly. Checkpoints also register signals from outside the cell. Three major checkpoints are found at the G1, G2, and M phases. For many cells, the G1 checkpoint, the restriction point in mammalian cells, is the most important. If the cell receives a go-ahead signal at the G1 checkpoint, it usually completes the cell cycle and divides. If the cell does not receive the go-ahead signal, the cell will then exit the cell cycle and switches to a non-dividing state, or the GO phase. Most cells in the human body are in that GO phase. Liver cells can be called back to the cell cycle by external cues, such as growth factors released during an injury to the organism. Highly specialized nerve and muscle cells will never divide. The cell cycle clock is regulated by cyclins and cyclin-dependent kinases. Rhythmic fluctuations in the abundance and activity of cell cycle control molecules pace the events of the cell cycle. These regulatory molecules are mainly proteins of two types, protein kinases and cyclins. Protein kinases are enzymes that activate or inactivate other proteins by phosphorylating them. Particular protein kinases can give the go-ahead signals at the G1 and G2 checkpoints. The kinases that drive the cell cycle are present at constant concentrations, but they also require the attachment of that second protein, the cyclin, to become activated. Levels of cyclin proteins can fluctuate cyclically. Because of the requirement for binding of a cyclin, the kinases are called Cyclin-Dependent Kinases, or CDKs. Peaks in the activity of one cyclin CDK complex, MPF, corresponds to peaks in cyclin concentration. Cyclin levels rise sharply during S and G2 phases, then fall abruptly during mitosis. Here we can see that fluctuation of the MPF activity in the cyclin. There's molecular mechanisms that help regulate the cell cycle. That MPF, or maturation promoting factor, or M-phase promoting factor, triggers the cell's passage past the G2 checkpoint into the M phase. MPF promotes mitosis by phosphorylating a variety of proteins. It acts both directly as a kinase and indirectly by activating other kinases. MPF stimulates fragmentation of the nuclear envelope, or the breaking apart of that nuclear membrane. And it also contributes to the molecular events required for chromosome condensation and spindle formation during prophase. MPF triggers the breakdown of cyclin, reducing cyclin and MPF levels during mitosis and inactivating MPF. The non-cyclin part of MPF, the CDK, persists in the cell in an inactive form until it associates with new cyclin molecules synthesized during S and G2 phases of the next round of the cycle. At least 3 CDK proteins and several cyclins regulate the key G1 checkpoint. Fluctuating activities of different cyclin-CDK complexes are of major importance in controlling all of the stages of the cell cycle. Research scientists are currently working out pathways that link signals originating inside or outside the cell with responses by cyclin-dependent kinases or other proteins. The M-phase checkpoint ensures that the kinetochores of all the chromosomes are properly attached to the spindle at the metaphase plate before anaphase. In this case, the regulatory molecule is not a cyclin-CDK complex, but a complex made up of several proteins. Once activated, the complex sets off a chain of molecular events that activates the separate enzyme, which cleaves the cohesions and allows the sister chromatids to separate. This ensures that daughter cells do not end up with missing or extra chromosomes. A variety of external and physical factors can influence cell division. For example, cells fail to divide if an essential nutrient is left out of the culture medium. Particularly important for mammalian cells are things called growth factors, which are proteins released by certain cells that stimulate other cells to divide. Researchers have discovered at least 50 different growth factors. Different cell types respond differently to specific certain growth factors or combinations of those growth factors. Platelet-derived growth factors, PDGFs, are produced by blood platelets. They're required for the division of fibroblasts in culture. Fibroblasts, which are a type of connective tissue cell, have PDGF receptors on their plasma membranes. PDGF molecules bind to these receptor tyrosine kinases, and this triggers a signal transduction pathway that allows certain cells to pass that G1 checkpoint and divide. In a living organism, platelets release PDGF in the vicinity of an injury. The resulting proliferation of fibroblasts helps to heal the wound. The effect of an external physical factor on cell division can be seen in density-dependent inhibition of cell division. Cultured cells normally divide until they form a single layer on the inner surface of the culture container. If a gap is created, the cells will grow to fill that gap. The binding of a cell surface protein to its counterpart on an adjoining cell sends a growth-inhibiting signal to both cells, preventing them from moving forward in the cell cycle, even in the presence of growth factors. Much like if you have a cut on your arm, that wound is going to grow closed from the outside edges to the center. Once it reaches the center, once those two sides are now touching, it signals the cells to stop growing. And so that connective tissue that has reformed, that scar tissue, will stop growing. Most animal cells can also exhibit anchorage dependence for cell division. The cells, to divide, must be anchored to some substratum, typically the extracellular matrix of a tissue. Experiments suggest that, like cell density, anchorage is signaled to the cell cycle control system via pathways involving plasma membrane proteins and elements of the cytoskeleton that are linked to them. Cancer cells exhibit neither density-dependent inhibition nor anchorage. Cancer cells have escaped that cell cycle control. So cancer cells divide excessively and they invade other tissues because they're free of the body's control mechanisms. Cancer cells do not stop dividing when growth factors are depleted. This is because cancer cell manufactures its own growth factors. It has an abnormality in the signaling pathway or has an abnormal cell cycle control system. The underlying basis of these abnormalities is one or more genetic changes that can alter the function of the cancer cell's protein products, resulting in faulty cell control. If and when the cancer cells stop dividing, they do so at random points, not at the normal checkpoints in the cell cycle. Cancer cells may divide indefinitely if they have a continuous supply of nutrients. In contrast, nearly all mammalian cells will divide 20 to 50 times under culture conditions before they stop, age, and die. Cancer cells, however, can become immortal. HeLa cells, if you remember back to biology, we talked about Henrietta Lacks. Those HeLa cells from a tumor are still reproducing in culture, and those initial cells were taken from Henrietta in 1951. The abnormal behavior of cancer cells begins when a single cell in a tissue undergoes a transformation that converts it from a normal cell to a cancer cell. Normally, your immune system will recognize and destroy those transformed cells. Cells that evade destruction proliferate to form a tumor or a mass of abnormal cells. If the abnormal cells remain at the originating site, the lump is called a benign tumor. Most benign tumors do not cause serious problems, and they can be fully removed by surgery. A malignant tumor, however, includes cells whose genetic and cellular changes enable them to spread to new tissues and impair the functions of one or more organs. An individual with a malignant tumor is said to have cancer. Cancer cells are abnormal in several different ways. They can have an unusual number of chromosomes, their metabolism may be disabled, and they can cease to function in any constructive way. Cancer cells can secrete signal molecules that cause blood vessels to grow towards the tumor. In addition to chromosomal and metabolic abnormalities, cancer cells can often lose their attachment to nearby cells and are then carried by the blood or lymph system to other tissues, and then start more tumors in new tissues. This is called metastasis. We say that a tumor has metastasized when it breaks free from its original tumor and is growing now somewhere else. Treatments for metastasizing cancers include high-energy radiation and chemotherapy with toxic drugs. These treatments target actively dividing cells. Chemotherapeutic drugs interfere with specific steps in the cell cycle. For example, Taxol prevents microtubule depolymerization, preventing cells from proceeding past that metaphase point. Side effects of chemotherapy are due to the drug's effects on normal cells. For example, nausea results from chemotherapy's effect on your intestinal cells. Hair loss results from its effect on the hair follicle cells. And susceptibility to infection results from its effect on the immune system cells. Researchers know a great deal about cell signaling pathways and how their malfunction is involved in the development of cancer through the effects on the cell cycle. Coupled with new molecular techniques such as the ability to rapidly sequence the DNA of cells in a particular tumor, medical treatments for cancer are becoming more personalized for each person's tumor. The development of cancer is a very complex process with many areas that remain yet to be explored. Perhaps the reason there are so many unanswered questions about cancer cells is that there's still so much to learn about the functioning of a normal cell. Here we can see a breast cancer cell. The tumor grows from a single cancer cell. It begins growing and it invades other tissues that are nearby. Most likely it's going to travel to the lymph system, invade the nearby lymph nodes, and then if it breaks free, it can metastasize or travel to another part of the body. And here we have the colorized SEM of a breast cancer cell.